4 billion years of Earth's history had already passed like a slow and ancient dream. The planet had changed many times and life had changed with it. What began as simple molecules inside warm oceans had grown into the world of tiny bacteria that covered nearly every corner of the planet. They harvested sunlight, consumed chemicals, and learned to thrive in places no complex organism could survive. But for more than 1 billion years, life remained small. Too small to see, too simple to build anything larger than a single cell. But the laws of evolution never stop. Given enough time, even the smallest, simplest forms of life face pressures that demand change. And deep within the oceans of the ancient Earth, change was about to begin. A new kind of cell was preparing to appear, one far more advanced than anything that had lived before. A cell that carried the potential for plants, animals, fungi, and eventually us. A cell with compartments, internal engines, and the ability to grow larger and more complex than anything the world had ever known. These were the first eukaryotes. To understand how extraordinary they were, we need to understand what life looked like before them. For billions of years, Earth was ruled entirely by prokaryotes, simple, single-celled organisms like bacteria and archaea. They were masters of survival, but limited by their simplicity. They had no nucleus, no mitochondria, no organized interior. Their DNA floated freely, their chemistry happened in open space, and their size was restricted by the very physics of diffusion. They could multiply fast, adapt fast, and spread across the world, but they could not become larger, nor could they coordinate millions of reactions with the precision needed for complex life. The early Earth did not need complexity, it only needed survival, and bacteria were the perfect solution. But evolution is never satisfied. As the planet changed, especially after the Great Oxygenation event, the environment placed new demands on life. Oxygen brought new chemical opportunities, but also dangers. Powerful energy pathways became possible, far more efficient than anything before. At the same time, oxygen also created toxic stress. Life had to innovate to handle it. Some organisms learned to use oxygen for energy, gaining more power per molecule than any species before them. Others hid in places where oxygen could not reach. But one path, one incredibly unlikely path, was the most transformative of all: a merger. Not in the human sense. Not a partnership or alliance, something much deeper. One cell swallowed another, and instead of digesting it, the two cells found a way to live together. Imagine an ancient world with no animals, no plants, no predators, just drifting microscopic life. And in that world, one archaeal cell engulfed a bacterium. Normally, this would end with the bacterium being broken apart and recycled as food, but something unusual happened. The engulfed bacterium kept living inside its host. More than that, it began supplying energy. It could take oxygen and convert it into power far more efficiently than anything else alive. The host cell, in return, offered protection and nutrients. This was not cooperation at first. It was survival. But survival turned into dependence. Dependence turned into integration. Integration turned into something entirely new. A single organism built from two lineages fused into one. This event created the ancestors of all eukaryotes. The engulfed bacterium would one day become the mitochondrion, the power generator inside every complex cell on Earth. Without mitochondria, complex life could not exist. Multicellular organisms would have been impossible. Brains would have been impossible. Animals, trees, insects, coral reefs, mushrooms, even the cells in your body, all impossible. This one event changed everything. But how rare was it? Life existed for billions of years before this merger happened even once. Many scientists believe it may have been the single most improbable step in the history of evolution. The creation of a nucleus, the organization of DNA, the internal skeleton of the cell, all of these came later. The defining moment, the turning point, was the merging of two very different forms of life into a single cell with unprecedented energy. Once this new cell existed, It unlocked possibilities the world had never seen. Larger size, more DNA, more complex chemistry, internal storage compartments, the ability to build structures, networks, and eventually tissues. But this transformation was slow. In the beginning, eukaryotes looked like nothing more than slightly larger microbes. They were not animals, not plants, just cells with better tools inside. Yet those tools would slowly reshape the entire planet. And so, somewhere around 2 billion years ago, the world entered a new chapter, the age of complexity, the age of innovation at the cellular level, and the age that would eventually lead to organisms that could swim, crawl, think, dream, and build civilizations. Eukaryotes spread like quiet pioneers across the oceans. They remained small at first, invisible to the naked eye, but they carried the blueprint for everything that would come later. Their mitochondria allowed them to harvest far more energy than their prokaryotic ancestors. Their internal membranes allowed them to isolate chemical reactions, to specialize tasks, and to begin organizing themselves into patterns shaped by evolution. And their DNA, now stored safely inside a nucleus, could grow larger, more complex, and more diverse. The stage was set for the most profound biological revolution in Earth's history since the origin of life itself. But evolution never moves in straight lines. The rise of eukaryotes was not a sudden explosion. It was a slow, patient climb. The early forms existed in a world still dominated by bacteria. They competed for space, for nutrients, and for survival. At times, they barely held on. But their advantages were too powerful to be ignored forever. It was only a matter of time before they began to transform the planet. In the quiet depths of Earth's ancient oceans, early eukaryotes drifted through waters rich with minerals, sunlight, and chemical potential. They were still humble organisms, far from the complexity of animals or plants, but they carried a new kind of internal architecture that set them apart from everything else alive. Their internal membranes folded and twisted, creating chambers where specialized reactions could occur. Their mitochondria worked like miniature engines, providing a steady flow of power that no prokaryote could match. They were tiny revolutions wrapped in delicate membranes. But evolution does not reward potential alone. It rewards the ability to survive. And the early eukaryotes still lived in a world where bacteria dominated every niche, every environment, every possible source of energy. Bacteria replicated faster, adapted faster, and could overwhelm entire ecosystems in days. A single eukaryotic cell, large, slow, and energy-hungry, seemed like a poor competitor in a world of fast, simple organisms. Yet, it was precisely their slowness and complexity that allowed them to evolve in directions bacteria never could. Inside their membranes, eukaryotic cells began developing systems of control. They built internal skeletons, microtubules, and actin filaments that acted like scaffolding. These allowed the cells to change shape, move more deliberately, and even engulf prey. This was something no prokaryote could do. They could hunt. For the first time in Earth's history, predation, true predation, began on a microscopic scale. Eukaryotes could extend their membranes outward like arms and swallow smaller organisms whole. This ability reshaped ecosystems. Instead of relying solely on chemical gradients or sunlight, these new cells could feed directly on other life. And with mitochondria powering these actions, they could afford to do it again and again. But hunting came with risks. Larger predators attract attention. They require vast amounts of energy. They must repair themselves constantly. To support these demands, eukaryotes evolved larger genomes and more sophisticated control machinery. DNA began to organize into chromosomes. Proteins evolved to regulate when genes were turned on or off. Cell cycles became more elaborate, tied to checkpoints and signals that ensured the organism divided only when ready. In these quiet changes, evolution was laying the groundwork for future complexity. The earliest forms of sexual reproduction emerged during this time as well. It was not sex in the way we understand it today, but a primitive form of genetic exchange. Cells fused, swapped chromosomes, and split again. This mixing of genetic material increased variation and accelerated natural selection. It also helped repair DNA damage, especially in an oxygen-rich world where oxidative stress could break genetic strands. For billions of years, life had relied solely on mutation and copying errors to introduce new traits. But sex changed everything. It allowed organisms to explore genetic combinations that would have been impossible otherwise. It allowed adaptation to accelerate, and it provided the fuel needed for evolution to take bold new steps. Meanwhile, the Earth itself was changing, the continents were drifting, volcanic activity was shaping the atmosphere, and oceans were undergoing cycles of nutrient enrichment and starvation. Iron, once abundant, began to vanish from seawater as it rusted and sank to the seafloor. This changed ocean color, chemistry, and biology. Life had to respond. Eukaryotes began experimenting with new ways to eat, new places to live, and new strategies for survival. Some evolved chloroplasts through another symbiotic event, where a eukaryotic cell engulfed a photosynthetic bacterium but did not digest it. This new internal partner allowed these cells to capture sunlight and convert it into energy. These organisms were the ancestors of all plants and algae. Once again, a merger between two cells created a path toward an entirely new branch of the tree of life. Still, this era was not yet the age of animals or forests. The world was a quiet place. Oceans were filled with tiny drifting eukaryotes and enormous populations of bacteria. No eyes would have seen these changes. No creatures would have sensed the shifting balance of power. But the groundwork was being laid for something extraordinary. The oceans were becoming laboratories of innovation. Evolution was running experiments everywhere. Some cells gained flagella and learned to swim more efficiently. Others flattened into sheets. Others developed protective walls. The diversity of form and function was increasing, even if the scale remained microscopic. One of the greatest innovations of this era was the cytoskeleton. This internal framework allowed eukaryotic cells not only to change shape, but to move materials inside themselves in organized ways. They could transport nutrients, build complex structures, and maintain stability while growing larger. Without the cytoskeleton, complex life would have been impossible. As the cytoskeleton evolved, so did intracellular communication. Eukaryotes developed signaling molecules that allowed them to sense their environment with increasing precision. They could detect chemical gradients, light, nutrients, and danger. They could respond with coordinated actions. For the first time, cells could behave in ways that hinted at future nervous systems and future organisms with integrated responses. And some eukaryotes evolved digestive vacuoles capable of breaking down prey in specialized compartments. Some developed peroxisomes to handle toxic chemicals. Others formed endoplasmic reticulum networks that folded proteins with remarkable efficiency. Each innovation expanded the possibilities of life. But one barrier still remained. As complex as these cells were becoming, they remained single units. To build larger organisms, they would need a way to adhere, communicate, and coordinate on a scale far beyond anything the world had seen. They would need to evolve systems of adhesion, signaling pathways, and mechanisms for cell-to-cell communication. Those systems were beginning to appear, but the world would not yet see their full potential, because Earth was heading toward one of the most extreme events in its history. A global transformation so severe that it would nearly freeze the entire planet. And this event would test eukaryotes in ways that would shape evolution forever. The age of the first complex cells was entering its next phase. In the slowly shifting oceans of the Proterozoic era, eukaryotes continued to evolve in ways subtle yet profound. They were not yet the mighty organisms that would dominate later eras, but they carried within them the seeds of future complexity. Their structures became more intricate, their chemistry became more refined, and their ability to manipulate energy gave them an advantage that quietly reshaped ecosystems. But evolution does not move in isolation. Every new adaptation changed the world around it, and every change in the world forced adaptation in return. As eukaryotes diversified, they began competing not only with bacteria, but with each other. Some became larger. Others became smaller and more efficient. Some evolved defensive spines or thicker cell walls. Others became faster hunters. The invisible battles of the microscopic world became more intense. And natural selection acted on every trait with relentless precision. The oceans, meanwhile, were undergoing their own transformations. Nutrient cycles shifted. The chemistry of the water fluctuated. Volcanoes released minerals and gases that seeped into the seas and influenced biology. The continents moved slowly, dragging shallow seas across their surfaces. Climate patterns changed as the atmosphere evolved, and with these changes came periods of boom and collapse, when ecosystems flourished and then starved. Eukaryotes faced these cycles with increasing resilience. Their mitochondria made them flexible, able to survive in fluctuating energy environments. Their membranes allowed them to store resources internally during hard times. Their ability to reproduce sexually allowed them to generate new combinations of traits when old strategies failed. These advantages were subtle, but they mattered. Some eukaryotes experimented with life in colonies. They formed filaments, sheets, or spheres made of dozens or hundreds of cells. Communication between cells became a necessity. Adhesion molecules evolved to keep cells connected. Chemical signals evolved to coordinate growth and division. These early colonies were not yet true multicellular organisms. If one cell died, the colony did not die with it. Each cell remained a fully independent unit capable of survival on its own. But this was a critical step toward complexity. To build animals, to build plants, to build fungi, life needed to move from cooperation to integration. Cells had to become dependent on each other. They had to specialize in roles so extreme that they could no longer live alone. This transition would take millions of years, but it began here, in these quiet colonial experiments drifting through ancient seas. Other lineages took different paths. Some single-celled eukaryotes became apex predators of the microscopic world, using flexible membranes and powerful internal machinery to engulf and digest prey. They developed sophisticated sensory systems capable of detecting vibrations, chemical trails, or tiny shifts in light. Their movements became more coordinated, their strategies more refined. The microscopic world was turning into an ecological battlefield, filled with hunters, defenders, scavengers, and specialists. For the first time, evolution began shaping behaviors, not conscious behaviors, but patterns of movement and response that allowed organisms to survive in complex environments. These patterns laid the foundation for the behavioral diversity that would later fill the animal kingdom. But the rise of eukaryotes also had a planetary impact. As algae and other photosynthetic eukaryotes spread, they pumped more oxygen into the oceans. This altered the chemistry of the sea floor, reshaped nutrient cycles, and changed which organisms could survive. Oxygen created new opportunities for those who could use it, but it also posed a lethal threat to organisms that could not adapt. Balance between oxygen-producing and oxygen-consuming life forms shifted constantly. At times, oxygen levels rose slowly. At other times, they stagnated or even fell. The atmosphere was in a state of long-term flux, driven by the interplay between geology and biology. Eukaryotes were not controlling the planet, but they were influencing it in ways no previous life form had. Yet, despite their growing influence, eukaryotes remained fragile. Their complex structures made them vulnerable to environmental stress. Their slow reproduction made them susceptible to extinction during sudden changes. They were resilient in some ways and delicate in others, and Earth was about to challenge them in ways far harsher than anything they had experienced. As the Proterozoic continued, the planet began cooling. Weathering of rocks drew carbon dioxide out of the atmosphere. Continents drifted into new arrangements that disrupted ocean currents. Volcanic activity shifted, and slowly, almost imperceptibly at first, global temperatures began to fall. Ice formed at the poles and crept outward. Snow accumulated in mid-latitudes and did not fully melt in summer. The planet began losing heat faster than it could gain it. A tipping point approached, one that would plunge Earth into a freeze so deep, so global, that it would be one of the most severe climate events in history. But we are not yet at that moment. Before the ice advanced across continents and oceans, the stage needed one more player, one more transformation, one more push that would reshape the balance of biology. That player was algae, not the seaweeds or kelps we know today, but primitive photosynthetic eukaryotes that drifted through the oceans as microscopic plankton. They were small, fragile, and often overlooked by predators, but they multiplied rapidly in nutrient-rich waters and could capture sunlight with remarkable efficiency. As they spread, they drew carbon dioxide from the atmosphere through photosynthesis and buried it in the ocean as dead cells sank to the seafloor. This burial of carbon acted like a planetary thermostat set to cool. Over millions of years, the Earth continued to lose greenhouse gases, and global temperatures kept falling. Eukaryotic algae did not know what they were doing. They were simply living, but their collective impact was enormous. Still, eukaryotes continued to diversify. New lineages emerged. Some formed shells made of silica or calcium carbonate, leaving fossils that would one day help scientists trace their evolutionary history. Others became early fungi, breaking down organic matter and recycling nutrients into the ecosystem. Others formed symbiotic relationships with bacteria that helped them survive harsh conditions. In this era, life was becoming more interconnected, more interdependent, and more dynamic. Ecosystems were becoming complex networks rather than simple chains. And eukaryotes were at the center of these networks, influencing the world in ways far beyond their size. But Earth was moving steadily toward a crisis. As temperatures dropped, glaciers expanded. Ice reflected sunlight, causing even more cooling. The oceans began to freeze at the surface. Carbon dioxide levels continued to fall. And eventually, the planet approached a threshold from which there would be no easy return. This threshold would lead to an event so severe that nearly every eukaryote would face extinction. An event that would freeze the oceans, cover continents in ice, and turn the planet into what many scientists believe was a near-complete snowball. Only the hardiest organisms would survive. Only the most adaptable would continue evolving. But before we reach that frozen world, we need to understand what eukaryotes had become by this time. They were no longer a single type of organism. They were an entire domain of life. They included predators, photosynthesizers, decomposers, symbiotic specialists, and colonial experimenters. They lived in shallow seas, deep oceans, coastlines, and even early freshwater environments. They were not dominant in terms of biomass or abundance. Bacteria still held that crown. But they were dominant in potential. Their cellular complexity allowed them to explore evolutionary paths that bacteria never could. They had mitochondria to generate powerful energy. They had nuclei to safeguard and organize large genomes. They had cytoskeletons that allowed dynamic movement. They had internal compartments that enabled specialization. All the ingredients for multicellular life were present. The world was waiting for the right conditions, the right pressures, the right moment. That moment was approaching. A global crisis would push eukaryotes to their limits, and only those that learned to endure the frozen earth would survive to spark the next great leap in evolution. The world did not freeze all at once. It crept into ice, step by step. Snowfall lasted longer each year. Glaciers crept down from the poles, inching across land that had once been ice-free. Sea ice spread outward from the polar oceans, thickening, reflecting more and more sunlight back into space. Each new patch of ice made the planet a little colder. Each degree of cooling allowed more ice to grow. Eventually, the Earth crossed a threshold. Ice reached the mid-latitudes. Then the tropics. Glaciers ground across continents, carving valleys and crushing rock into powder. Sea ice thickened until it sealed large portions of the oceans away from the sky. The Earth had become, in many ways, a snowball. This was the Snowball Earth event, or rather, one of several. It was not a moment, but an era, an episode in which the planet was locked in deep freeze for millions of years. To most life on the surface, it was catastrophic. Sunlight no longer reached the ocean depths as it once had. Photosynthesis stalled beneath thick layers of ice. Nutrient cycles slowed to a crawl. The world that had once been a cradle of slow but steady biological innovation was suddenly a hostile, frozen prison. For eukaryotes, whose existence depended on delicate balances of temperature, light, and nutrients, this was a trial by ice. Yet even in a world that seemed dead from afar, life endured. Life retreated into refuges, small, scattered pockets where conditions remained mild enough to allow survival. Around volcanic activity, where heat seeped from the crust, liquid water persisted beneath the ice. Cracks in the sea ice allowed thin beams of light to reach patches of ocean where photosynthetic organisms could still function. In deep-sea hydrothermal vents, where sunlight had never mattered, bacteria and archaea continued their ancient chemistries. Eukaryotes that could tolerate darkness, cold, and scarcity clung to existence in these refuges. In these hidden pockets, evolution continued. The stresses of Snowball Earth selected for resilience. Organisms that wasted energy could not survive. Those that could enter dormant states, repair damage, or endure long periods of starvation had an advantage. Cellular systems for controlling metabolism became more sophisticated. DNA repair mechanisms improved. Membranes adapted to lower temperatures, becoming more flexible in the cold. The extreme environment acted as a sieve, filtering out the fragile and preserving the hardy. The eukaryotes that endured this era were not just survivors, they were future ancestors. As the world remained frozen, geological processes continued beneath the ice. Volcanoes emitted carbon dioxide into the atmosphere, but with the ocean sealed off, much of this CO2 could not dissolve into the water. Instead, it accumulated slowly in the air. Over millions of years, the planet's greenhouse blanket thickened. The heat trapped by this carbon dioxide gradually began to overpower the cooling effect of the reflective ice. Temperatures rose, almost imperceptibly at first, but steadily. Then at some tipping point, the ice began to melt. What had taken millions of years to freeze began to thaw more rapidly. Glaciers retreated. Dark ocean water was exposed again, absorbing sunlight instead of reflecting it. Runoff from melting ice carried enormous amounts of ground rock and nutrients into the seas. The world shifted from a frozen silence to a roaring flood of change. For eukaryotes, the thaw was both a relief and a challenge. When the oceans reopened, they were full of nutrients, phosphates, nitrates, and trace minerals washed from the land by massive glacial meltwater. These nutrients fueled explosive growth in photosynthetic life. Algae and cyanobacteria bloomed, turning the oceans green and teal as they captured sunlight and multiplied. This post-snowball world was wild and dynamic. Ecosystems that had been confined to refuges now spread across vast open waters. Eukaryotes had new space, new resources, and new opportunities, and evolution, given such abundance, responded with creativity. Algae diversified into many forms: filamentous, spherical, colonial. Some remained microscopic plankton. Others began to form larger, more complex bodies. Predators returned to the open oceans, feeding on the blooming algae. Single-celled hunters swam through the water, tracking prey by chemical gradients and subtle currents. Some used flagella to propel themselves. Others glided using cilia. All of them operated with a sophistication that ancient bacteria could never have matched. The thaw after Snowball Earth did more than restore life, it accelerated it. In this new world, eukaryotic cells continued their march toward multicellularity. Colonies that had once been loosely bound began to develop stronger adhesion mechanisms. Cells evolved proteins specifically designed to hold them together. Instead of drifting apart after division, daughter cells remained joined in stable patterns: chains, clusters, sheets. Cell communication became more complex. Chemical signals traveled between neighboring cells, coordinating growth and response. Some cells started to suppress their own reproduction and instead supported the colony as a whole. Others specialized in movement or nutrient absorption or reproduction. This division of labor represented a profound shift. Life was no longer just about individual cells trying to survive alone. It was about collectives of cells acting as a single entity. True multicellularity was emerging. Early multicellular organisms were still simple by modern standards. They had no organs, no bones, no hard shells. They were delicate aggregates of cells, floating in water, anchored to rocks, or creeping slowly along the seafloor. Yet they were radically different from anything that had existed before. They could grow larger than single cells. They could form shapes. They could coordinate internal processes across many cells at once. To build such organisms, evolution had to invent new solutions. It had to create ways to control which genes were active in which cells. It had to create systems that allowed cells to know where they were in a body, whether they were on the outside, inside, top, or bottom. It had to evolve developmental programs, sequences of gene expression that unfolded like blueprints as an organism grew. These innovations did not appear overnight. They accumulated slowly over millions of years. In countless lineages, most did not leave fossils behind, but their legacy can still be seen today in the genetic machinery that controls the development of every plant and animal. The rise of multicellular eukaryotes changed ecosystems once more. Larger organisms shaded smaller ones from light. Colonies created new surfaces for other microbes to live on. Organic matter accumulated in new forms when these larger bodies died and decomposed, feeding fungi and bacteria that specialized in breakdown. Food webs became more layered. Instead of a simple chain of producer and consumer, there were now multiple steps. Primary producers, grazers, predators, scavengers. Nutrient cycles became more complex and efficient. Oxygen played a central role in supporting this complexity. Larger organisms needed more energy. More energy required more efficient respiration. And oxygen was the perfect fuel. The very gas that had once driven mass extinction had now become the backbone of thriving, intricate ecosystems. As multicellular eukaryotes spread, the Earth's continents continued to move. Supercontinents formed, broke apart, and formed again. Shallow seas flooded continental margins, creating ideal environments for new life. Tides, waves, and storms mixed nutrients through these waters, stimulating further productivity. Meanwhile, the ozone layer high above the planet protected these ecosystems from ultraviolet radiation. For the first time, life had safe access to sunlit, shallow waters near the surface. These environments were nurseries for innovation. It was in these coastal habitats that some of the earliest large multicellular organisms likely arose. Simple, soft-bodied beings that looked like fronds, discs, or quilted shapes lying on the seafloor. They were the distant precursors of the Ediacaran biota and later the Cambrian explosion. But at this stage in the story, they were still experimental forms. Evolution was testing the possibilities of multicellular architecture, how to build, how to shape, how to persist. Through all of this, eukaryotic cells remain the fundamental unit of complexity. Every multicellular organism, no matter how large or simple, was made of them. Each cell carried mitochondria gained from that ancient symbiotic event. Each one stored its genetic material in a nucleus. Each one depended on elaborate systems of protein folding, signaling, and structural scaffolding. The rise of eukaryotes had not been a single moment, but a drawn-out transformation. From the first symbiotic merger to the first true multicellular bodies, hundreds of millions of years had passed. But now, with a thawed planet, rising oxygen levels, and a biosphere richer than ever before, that transformation was nearing a turning point. Life was ready to take the final steps toward visible complexity. The world stood on the threshold of an era in which evolution would paint the oceans with shapes, colors, and forms that had never existed before. The foundation was laid. The stage was set. What happened next would forever change what life could be. The world after the Great Thaw was not a quiet place. It only looked calm on the surface. Beneath the gentle sway of warming oceans, evolution was accelerating with a force it had never known before. Every coastline, every shelf sea, every patch of shallow water was becoming a laboratory, a place where complexity was being tested, stretched, broken, and rebuilt again. For billions of years, life had lived in a microscopic world, a world too small to see, too simple to shape the planet in any visible way. But now, with eukaryotes thriving and oxygen slowly rising, the oceans were beginning to think bigger. Cells that once drifted alone were starting to gather, cooperate, and experiment with new forms of existence. They were no longer simply surviving, they were exploring possibility. Most of this evolution happened in silence. A single mutation here, a tiny shift in behavior there. But over millions of years, those small changes stacked like pages in a book. And the story they told was something entirely new. The emergence of organisms that were not just cells, but societies of cells. In the shallow waters, colonies formed sheets, branches, and ribbons of living tissue. Some grew outward, catching sunlight like miniature mats. Others grew downward, anchoring themselves to sediment. Still others formed spherical or tubular shapes, hollow on the inside, filled with fluid and purpose. These early multicellular structures didn't yet resemble animals or plants. They were experiments, flexible blueprints, testing the physics of size, shape, and cooperation. Some divided labor between cells. Outer layers protected, inner layers processed nutrients, hidden layers reproduced. It was primitive, but it was a beginning. Every branch of complexity was being sampled. Nature was running millions of prototypes at once. Meanwhile, predators were changing the rules entirely. Single-celled hunters had always existed, but the thawed oceans made them fiercer. Some evolved stronger pseudopods for grabbing prey. Others developed intricate feeding grooves. Some even began secreting chemicals to stun or dissolve other cells before consuming them. Predation forced prey to adapt fast. It pushed organisms toward new innovations: speed, awareness, coordination, armor, toxins. This arms race played out at microscopic scales but produced macroscopic consequences, because each adaptation nudged life closer to forming true tissues, true bodies, true organisms. And every step forward depended on oxygen. Not in huge amounts, not yet, but just enough to power these new strategies. Enough to give evolution the fuel it needed to reach beyond simple existence. Deep inside these eukaryotic cells, the machinery of complexity kept refining itself. New gene regulatory networks formed, systems that could turn entire clusters of genes on or off, like a switchboard. In older life, a mutation might alter one protein, but now, a single change could alter whole developmental pathways. Bodies could change shape more easily. Cells could specialize more deeply. Evolution gained flexibility, and with it, speed. The cytoskeleton also grew more advanced. Cells could move materials with precision, rearrange themselves, stretch or curl or divide in elaborate patterns. This internal scaffolding acted like a quiet architect, helping cells build structures greater than themselves. Life was preparing for its next leap, even if it didn't know it. With more oxygen and nutrients swirling through the seas, the stage set itself for the first large organisms. They appeared not suddenly, but gradually, soft-bodied, simple, and delicate. Some looked like elongated ovals pressed into sand. Others resembled fronds, branching like slow underwater flames. Some were quilted, divided into repeating segments, like arcs or leaves. Others grew like discs on the seafloor, leaving ghost-like rings behind. These organisms are known today as the earliest macroscopic eukaryotes, the precursors to the Ediacaran biota. But in their day, they were just another experiment with size and shape. They had no eyes, no mouths, no muscles. They absorbed nutrients through their surfaces, letting currents feed them. Their bodies were soft enough that only the most perfect conditions preserved them. And yet they spread, thriving quietly across ancient seafloors. For the first time in Earth's 4-billion-year history, an organism could be seen without a microscope. That alone was revolutionary. In the water around them, countless microscopic species still dominated the ecosystem. But now there was a new layer of life, one that changed the flow of nutrients, the movement of water, the chemistry of sediments. Life was no longer just reacting to the environment. It was beginning to shape it. Meanwhile, in the microscopic world, another transformation was underway. Some eukaryotic cells began forming permanent associations with others. Small cells attached to larger ones. Colonies practiced coordinated movement. Cells communicated chemically, sending signals to regulate growth and behavior. These were the early foundations of tissues, collections of cells that worked together instead of acting alone. This cooperation deepened into reproduction as well. Sexual reproduction, which had originated earlier in eukaryotes, now spread widely. It mixed DNA, sped evolution, and generated variation faster than ever before. It also created stable life cycles, linking generations through developmental stages that repeated like ancient rhythms. All of this, cellular cooperation, increasing size, rising oxygen, predation, and genetic innovation, was pushing life toward a threshold, the threshold of animals. But evolution wasn't rushing. It was patient, sculpting complexity one tiny step at a time. For tens of millions of years, the oceans were filled with these strange, soft-bodied forms. Some grew tall like underwater feathers. Others spread flat like carpets across the seafloor. Some floated. Some crawled slowly. Some simply clung to the sediment and filtered passing nutrients. They formed the first ecosystems, larger than microscopic films. These communities began modifying the ocean floor, stabilizing sediments, altering flow patterns, and creating pockets where new species could evolve. Life was building its own stage. By around 600 to 580 million years ago, the world had changed enough to support something more. Oxygen levels were high enough to allow sustained growth. Temperatures had stabilized, nutrients flowed reliably, and the genetic machinery inside eukaryotes had reached a point where it could build complex bodies with specialized tissues. The toolkit for animals already existed. It was just waiting for the right moment to be used. That moment was coming soon, sooner than evolution had ever moved before. A new era of life was approaching, one filled with creatures that could move with intention, sense their surroundings, hunt, hide, grow, and reproduce in ways never seen before. The Ediacaran period would reveal them. The Cambrian would unleash them. But right now, in the fading quiet before the dawn of animal life, the world was poised on the edge of transformation. The rise of eukaryotes had done its work. It had created the architecture of complexity. It had given life the tools it needed to become magnificent. And in the deep time between the last great ice ages and the first true animals, the oceans held their breath, because the next chapter wouldn't just change life, it would change the rules of evolution forever.